Good afternoon, everyone. I'm very pleased to be here in Zoom again for one more webinar. As you may remember, the idea of organizing these webinars was the result of the pandemic crisis, being a way of sharing knowledge and discussing relevant topics in the food area, but also a way of staying connected with our fellow colleagues. We received a very good feedback for the previous webinars, and in fact, due to COVID, these new practices and a more digital age is here to stay. And therefore, the Food Chemistry Division, with the support of EuChemS, will continue these events, aiming to organize 4 webinars this year. The situation with COVID is now much better, but unfortunately, humanity now faces a new crisis with the violent invasion of Ukraine. Science is made up of collaborations that allow advances and new discoveries, independently of country, race, religion, gender, personal choices. Science has no flags or geopolitical borders. Thus, as chair representing the Food Chemistry Division, I would like to express our solidarity to all that are experiencing these dramatic happenings. Our thoughts, wishes, and for the ones who believe, our prayers are with Ukrainians and with all of those who defend freedom, democracy, human rights, and of course, science. And thus, I would like for some brief moments to share with you the statement of EuChemS regarding this invasion. So thank you all so much for joining us. And now let's proceed with our webinar. And I give the floor to Doris. Thank you.
Thank you, Joana, for the introductory words and the real important statement with respect to the invasion of the Ukraine. So, but now let's come to science, and it's my great pleasure to introduce to you the speaker of today, Professor Christopher Elliott, who will give us today a talk on the fighting of global food crime with analytical chemistry. So let me introduce Professor Elliott a little bit. So he is currently Professor of Food Safety and founder of the Institute of Global Food Security at Queen's University, Belfast. So he is an absolute expert in the field and has published close to 500 peer-reviewed articles. And in addition to his position at the University of Belfast, he is also a visiting professor at the China Agricultural Institute in Beijing, the Chinese Academy of Sciences. So he has received a whole panel of fellowships and prizes, and this is just a too long list to read it now today. So on this list, among others, he is an elected member of the Royal Irish Academy, and he's vice president of the Chartered Institute of Environmental Health. He was nominated 2021, and many other prizes. And with that, I don't want to let you wait too long for Chris. So Chris, it's my great pleasure to have you here in the webinar today. We would have enjoyed you in an on-site congress, but if not so, it's our pleasure to hear you at least online. So the floor is yours, Chris.
Many thanks for the very kind invitation from the European Chemical Society to give a presentation. And thank you, Doris, for a very warm welcome. It's a pleasure to be talking to you. And what I'm going to do is really now try to talk about some of the aspects around some of the research that myself and my colleagues have been undertaking for quite some time in terms of thinking about food chemistry and actually how we can use food chemistry to fight and combat what we believe is a very, very serious and growing problem right across the world. Now, this is a slide I always show to all of my new students at Queen's University Belfast. And I tell them they've got 10 minutes to memorize that slide. They always look scared. And the reason they look scared is it is so complicated. Our global food supply system is unbelievably complicated, but this is how we set out to feed nearly 8 billion people on our planet 365 days a year. And our system has evolved over many centuries, but particularly over the last 2 or 3 decades. And with such a complex system comes vulnerabilities. Things can go wrong. Some of those things can go wrong by accident, but some of them can go wrong by design. And what I'm going to talk to you today is about some of the things that people do to perpetrate criminal activity in our global food supply system. I have another lecture actually, and I call it Why I Invented Food Crime. Because actually, I came up with the terminology back in, my goodness, about 2013, 2014, to try to explain to people the amount of criminal activity that we do have locally, nationally, and internationally. And I often try to describe and explain this by using some nice maps of our world. So this map shows the major trading routes of agriculture and food commodities across the world. And I think you can see it's complex again. And actually Europe sits pretty much in the epicenter. Europe is the world's biggest trading market for food. Here's another map of our world. And you can see the trading routes are pretty much exactly the same, but this is the world's trading routes of organized crime. This is about firearms, it's about drugs, it's about people trafficking, smuggling, counterfeiting. And the routes and pathways are very much the same. And what I have observed over the past 5 to 10 years is there is a much closer connection now between organized crime and our global food supply system. So if I give you a statistic, is there's now more money made in food crime than there is in the whole heroin trade in the world. So we've got drug cartels in Central and South America, triad gangs, mafia in Europe, all becoming more and more evolved because so much money can be made. It's estimated that about 50 billion euros is made each year in terms of crime in our food system. Now, how is the crime perpetrated? And I divide it into 3 kind of clear sections. The first is we call substitution fraud, where you replace something of high value with lower value. And the European horse meat scandal was a great example of that. We also have a huge amount of fraud in olive oil in the world, and the substitution fraud there is replacing virgin or extra virgin olive oil with standard olive oil, or actually other vegetable oils. So again, a classic example of substitution fraud. Then we have what I call addition fraud, and that's where some pretty nasty people will add things to our food to make it seem that there is an increase in value. We do a lot of work in herbs and spice fraud, and I'm going to give you some more details of that a little bit later on. But the ability to add things to spices to make the color more intense is a very, very common way that fraud is perpetrated. And then we have the massive melamine scandal in China, where melamine was added to milk, particularly in infant formula, to give it a perceived higher level of protein. So that's addition fraud. And then we've got the collection of what we call false claims fraud. Because whenever we walk into a shop, a supermarket, a cafe, or a restaurant, what we do is we make choices, we make food choices. The first obvious choice is, what do we like to eat? The second choice is, how much does it cost? But then we look at other attributes of food. Where did it come from? Is it welfare friendly? Is it organic? Is it fair trade? And all of those things are open to cheating. Now, I have been investigating food fraud or food crime for a very, very long time. In fact, my first court case was in 1992, which is, I can say it's my 30th anniversary of fighting fraud. And what I can say is, those people who try to perpetrate fraud will try every particular way. If you try to build a wall, they'll go under it, go over it, go around it, or just break the wall down. Now, what I'm going to do is give you a few case studies, things that we have investigated in terms of fraud, myself, my research group, and one that we are currently investigating. We actually started quite locally. Now, how do we get this information about fraud? And I use lots of very clever ways I crawl the web. I've got intelligence networks. This particular example started when somebody at a football match came up to me and told me that there was fraud happening in the oregano trade in the UK. And this was my first exposure to fraud in herbs and spices. And what we decided to do was we would investigate oregano, or oregano, as some people call it, to find out if the fraud was really happening. Now, for all of those people on the webinar today, and I can see we've got 125 people registered, which is really good. I would say the vast majority of us in our kitchens at home will have one of those little jars there. It will contain oregano, and we will use it to add to our food and so forth. But also, oregano is used in many, many different types of foodstuffs, in processed foods and ready meals, to add the flavor to it. So when you look at this slide, the oregano plant is on the left-hand side, and it's very easy to tell if it's oregano or not. You can just look at the structure of the leaves. But how oregano is traded is through that picture in the middle. It's the dried, crushed product. And then those are the things that end up in those little jars or in food service. So here's a picture of 4 different types of oregano obtained from different parts of the world. Okay, and you can see slightly different colors. But then if I tell you, actually, that's not oregano, that is myrtle leaves. And when we looked at the way that science was used to detect fraud in oregano, I was really quite surprised, because it was based on looking down a microscope that had been developed about 300 years before, to look at the different shapes and colors and sizes of the leaves. And we thought, my goodness, there must be a much better way to think about measuring fraud in different types of food. And this is where we've come up with our concept about fingerprinting, the fingerprinting of food, to try to identify the species of the food, the breed, where it came from. Is it from sustainable production methods? Is it from capture methods? So this fingerprinting idea is, we will take a measurement and the fingerprint will say, yes, this is genuine, or the fingerprint will say, no, this is fraud. And we also think it's like a traffic light system. We test. If it gets a green light, everything's okay. If it gets a red light, there's a strong suspicion of some form of fraud. Now, we love 2 particular techniques in my research center. We love molecular spectroscopy and we love mass spectrometry. So a lot of our applications are around spectroscopy and mass spectrometry. And for the spectroscopy, we will use Raman, near infrared, FTIR, many different types. So in this particular example, it's about using FTIR. And what we were doing was, we would scan Oregano, we would scan those adulterants like olive leaves and myrtle leaves to look at the picture. And you can see the scans are relatively simple. But whenever you start to apply chemometrics, and chemometrics is the statistical analysis of chemical data, we can start to see that we can produce clusters of results. And in that diagram on the right-hand side, you can see a cluster of results for oregano, for cistus leaves, and so forth. So it's actually very, very easy to tell the difference between what is genuine oregano and what is olive leaves. And then we start to develop models where we will look at 10%, 20%, 30%, 40% adulteration to see if we can still discriminate against genuine products from fake products. So in terms of our work on oregano, based on our cross-validation, we were able to get the result right about 95, 96% of the time. That's pretty good science. And what we were able to say is we had developed a low cost, a rapid way of identifying whether oregano was genuine or whether it had been adulterated. But 95% seems like a good number. But from my experience, if you ever try to bring a prosecution to a legal court, if you try to revoke a contract based on 95% results, you will be in big trouble, because that basically means 1 in 20 times your analysis is giving you the wrong result. So we then look at what we call the second tier of testing. Spectroscopy is tier 1, let's do it quickly, but our second tier is much more around advanced scientific technologies and particularly the science of metabolomics. So what we would do in terms of metabolomics, we would screen the entire metabolome of different types of foods, and we would look for the phenotypic biomarkers, those biomarkers that said, this will show you it is genuine oregano, or sometimes these biomarkers will show you that it is an adulterant. This untargeted analysis, and we would generally do in platforms using time of flight mass spectrometry, and we would get this complex fingerprint of all of the metabolites. But again, then what we would do is the chemometric analysis to say what is genuine and what belongs to adulterants. Whenever we apply this type of advanced chemometrics in mass spectrometry, we can then get beautiful statistical models developed. So on the left-hand side of the slide, we can see there is the genuine oregano, and actually you can see there's 2 distinct clusters there. One is European oregano, one South American oregano. So not only can we tell it's oregano, we can tell what continent it came from. And then all of those samples of oregano that sit outside that cluster have some form of adulteration built into them. Now, what we did is, like all good scientists, like all good researchers, we published our work in a nice journal, saying we had developed this comprehensive strategy to detect the fraudulent use of herbs using oregano as a model. Then what we decided to do was a survey, and we did a survey, small survey of oregano that was present on the marketplace in the UK and Ireland, either from supermarkets, shops, or on the what could be bought over the web. What really surprised us was that about 25% of every sample that we tested had some form of adulteration. So 1 in 4 samples of oregano on sale in the UK market was adulterated. Now that was a much bigger number than we had suspected. So what we did was we published our work, and then we were inundated with people from different parts of the world saying, will you test our oregano for us as well? We teamed up with a consumer organization in Australia. They sent us 12 products of Australian oregano. We tested those in our 2-tier system. And guess what? It wasn't 25% fake. It was 65% fake. That most of the oregano on sale in Australia had some form of adulteration. And based on our work then, some of those companies that were selling that adulterated material were prosecuted. They were taken to court. And remember what I talked about, that confidence of your analytical test, and we were able to prove in court that actually the samples were truly adulterated. And this just didn't happen in the UK, Ireland, in Australia. It's happened in many different countries across the world. This was our investigation in Norway, and again, we found quite a lot of fraud there, and various companies have been prosecuted for fraud in oregano. We've collected a lot of different samples from different parts of the world, and basically wherever we look for fraud, we find it, but at different levels. So if anybody is interested in saying, I wonder is my oregano on sale in my country fraudulent, you contact me, we will test it and we will let you know, and your data will be added to our graph. So globally, we found about 25% of all oregano is adulterated. Now, another part of our investigation was, particularly in Europe, what we know is a lot of the oregano is grown there and processed there. Then we started to do what's called a mass balance check. How much is produced and how much is sold? Now, I got the original tip-off for oregano back in 2014, and we looked at the mass balance. We found that about 12 million tons of oregano was produced and about 14 million tons was sold. Wow, that's what you call great productivity. Cheating. After our investigation was over, we went back and did a mass balance, and guess what? The mass balance is perfect. 15 million tons produced and 15 million tons are exported. So this really shows you the impact of having good analytical tests to detect fraud. Another very interesting thing, in Turkey, in their industry, they all know not to cheat because they know a professor from Queen's University will find they're cheating. Now, what I want to do now, move on to another example of how we've looked at the innovations of food fingerprinting. And again, this time it's using advanced mass spectrometry, time of flight mass spectrometry. But we've taken it one step further. What we have done is, to the front end of the mass spectrometer, we have added what's called the REAM system, the rapid evaporative ionization mass spectrometry. So basically how that works is we have a laser which burns our sample. Then the smoke that's produced from the sample gets injected into the mass spec, and within 5 seconds, we get our analytical result. No sample preparation, 5 minutes analytical time. Quite unbelievable, the power of REAMS-based mass spectrometry. And we refer to this as ambient mass spec. And it's not only REAMS we use now, but we use many different front ends to avoid the need for sample preparation. Now, the example I want to talk to you about is fish fraud, which is colossal right across the world. We looked at what the current state of the art was, and that was generally using some form of gel technique, which would measure bands of DNA or measure protein bands. And then from that, you could tell whether the species was correct or incorrect. The technique is expensive and it takes quite a lot of time. So what we decided to do was, could we use REAMS to rapidly detect speciation of fish? Now, in this particular project, we worked on 5 different species of whitefish: cod, coley, haddock, pollock, whiting. They all look virtually identical. Actually, genetically, they're very closely related. And what we wanted to know, using a fingerprinting technique, using ambient mass spectrometry, could we tell them apart? Now, our fingerprinting this time was around the fatty acids and the phospholipid profiles that were present in those fish samples. So we tested many different samples of the different species of fish to get the lipid profiles. And even from my slide there, you can see the lipid profiles are really very different, even though genetically these fish are very common. We did the same type of chemometric modeling, statistical analysis, and it was really very, very easy to separate those species of fish. Very, very easy. So within 5 seconds of testing a sample of fish, we could tell if it belonged to one of those 5 species. And actually, since then, we have added many more different species of fish to our model. So we call this the eye knife technology, where we can take a sample, we can burn it, and within a few seconds, without any type of sample preparation, we can get our analytical result produced. And our accuracy using reams sits at about 99%. So it's a very, very powerful technique. Now, whenever we were doing this particular project, I had a fantastic PhD student. His name's Connor Black, and he was doing all of this work. And actually, he did the work so quickly, it worried me because he became too confident. So I told him, Connor, this isn't enough. I want you to go and take all of the data that you have generated, doing all of the chemometrics. Tell me something else about these fish. And what he was able to do just by analyzing the data, we could tell the difference between a species of fish that came from a trawler, and we could differentiate it from a boat that was using line caught fish. Now, often when you walk into a supermarket, the claim will be it's line caught or pole caught, and we pay more money for that. Yet using the lipidomics approach, we could quite easily tell what came from a trawler and what came from a line. And then you think, how is that possible? The same species of fish, what was the difference? And our hypothesis is that it's the catch method. When fish are caught in a trawler, they tend to die from suffocation. When they are caught on a line, they tend to die from exhaustion from fighting. And what we're doing is we're picking up different stress pathways. So what we were able to do was, I call it the 11 sins of sea fish. And there are 11 different ways you can cheat in sea fish, that by using one analytical technique, you could measure 2 different types of fraud: speciation fraud and catch method fraud. Then I had a wonderful postdoctoral researcher join my group from India. And I said, okay, Niladri, you're going to go from 2 sins to 3 sins. So we gave him the challenge of saying, could he distinguish the species of the fish? Could he tell the geographic origin? And could he tell the production method of the shrimp in a single untargeted metabolomics approach? So here are the 5 most common, most popular types of varieties of prawns. You can see their genetics are different, they're produced in different countries and so forth. And we set out to collect a lot of these samples of shrimp and again, apply them to our reams technology. And again, look at the separation between those species, quite incredible. So within a few seconds, not only could we tell the shrimp, we could tell the country that it came from, and we could tell the production method very, very quickly. So you take a sample, you burn it, and within 2 or 3 seconds, on the right-hand side, that's what our software will say, that it is a king prawn from Thailand. What about that for science? Because there's so much cheating goes on in the world of prawns and shrimps. Now, the last case study that I want to talk to you about is something that we're doing now, right now. And that's about looking at the source of soya. Can we tell where our soya comes from? Now, the reason is soya is a very, very controversial feed and food at the moment. A huge amount of soya is produced. A lot of it goes into feeding cattle. Some of it goes into food. And there are a lot of concerns that a lot of deforestation, particularly rainforest deforestation, has happened in Brazil to clear room to produce more and more soya. So our hypothesis is that we could use analytical chemistry, not only to say what country did the soya come from, We want to say we can tell what region of the country it comes from. And the third thing, we can tell if the soya has been grown on land that has been recently deforested. So this whole topic of soya, it is very controversial. And now many, many different companies and supermarkets are saying, we are only using sustainable soya, blah, 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 without any evidence that they actually are using sustainable soya because of the complexity of those supply chains. And what we're now finding is that more and more land, particularly in Brazil, is being cleared to produce soya. So how can everybody claim to be using sustainable soya when actually you don't know? The statistics that we got from the UK is we can only track and trace about 20% of all of the soya that's used in the UK. So 80% we really don't know where it comes from on the production techniques. So what we've done is we've formed some beautiful partnerships with some of the great NGOs of this world, the World Wildlife Fund, 3 Keel, the Round Table on Sustainable Soya. And what we're doing is we're now in the midst of collecting many, many samples of soya from the 15 countries in the world that produce soya. If you happen to be listening from any of these countries, I've got a request for you in a little bit of time. So far, we've collected about 500 samples of soya from 13 different countries. Recently, we've signed wonderful agreements in North America, particularly in the United States and Brazil, for samples of soya to come from there. So we are building up our samples to have our database to do our analytical chemistry on. Where are we doing this chemistry? Well, this is just part of the technology center that myself and colleagues have built at Queen's University over the past 10 years. It's unbelievable. We have wall-to-wall spectroscopy and mass spectrometry. This is what we call Adjuvant Ally. So we have many, many different platforms who have been supplied to my research center from Adjuvant. So we have a very, very strong partnership. And we are using a lot of these platforms in our soya project. And what we are doing is these are our workflows now. We're thinking about looking at both organic and inorganic analysis to look at the footprint, the fingerprinting of volatile compounds, of non-volatile compounds, and doing elemental analysis. Elementomics, I'm now told it's called. One of my PhD students told me that a few weeks ago. It was a new word for me, but anything we publish doing elemental analysis now is elementomics. We're using GCMS, we're using time of flight, and we're using ICPMS, so different types of analytical platforms. It's really important when you start to develop your workflows is they are simple to use, and the more simple the better, because they're quicker, they are lower cost, and the ability to transfer those methodologies from one laboratory to another can be greatly enhanced by keeping things simple. So this is the type of workflows that we have developed for GCMS and starting to produce some beautiful results. Here are some preliminary data where, again, what we're starting to do is produce these fingerprints, the fingerprints of volatile compounds. Then what we can do is, using a lot of different chemical databases, we can start to identify what is the molecule that is specific to that particular soya that has come from a particular region of the world. And on this, we can see we've analyzed samples from Brazil, India, Ghana, Italy. And we are starting to pull out different markers of those particular countries. We're also now looking at our untargeted workflow using ICPMS. And again, it's a very, very simple standard workflow to help us, to encourage us to be able to transfer the methodology. The same is true for our LCQ-TOF, very, very simple. And when it comes to the idea about workflows, actually one of the things, like many other laboratories, we had to close down for a little bit of time because of COVID. I gave all of my researchers different projects. One of those projects was to look at dilute and shoot and think about actually redefining dilute and shoot. What does it really mean? What is the evolution of the technique, and how can it be applied to food analysis in terms of using GCMS and LCMS? And now that publication really has brought together all of our thinking about the workflows that we have developed. I would love you to download this. I would love you even more, of course, to cite it, even if you like it or don't like it, because we love our work to be cited. Now, where do we sit in our soya project? We're now sitting, collecting our hundreds of samples. We are now starting to apply them to different types of methodologies. We're working with lots of different stakeholders in terms of trying to get more and more samples into our laboratory to produce our fingerprints of all of those different biomarkers. So what I'm going to say is that what we formed is a coalition. We have a phenomenal coalition of companies, local, national, international, who all use soya. And they all want to know where their soya is coming from. Is it sustainable? Is it coming from a country which is linked to deforestation? And can we actually detect in those samples evidence of deforestation? And then again, that is about looking at the profiles. And we have a number of different hypotheses about how we will be able to identify continent, country, region linked to deforestation. So if you have any interest at all in joining our coalition, please email myself or my colleague Brian Quinn, who is leading on all of the mass spectrometry work. We really want to make this a global project. And also, when we develop our methodologies, we're going to give them away to anybody who wants them free of charge. We will help you get your methodologies running on your own instrumentation so you can do the same checking about supply chains coming into your own countries. That's my invitation to you. Now, if I asked the 125 people on this call, what is that animal? Everybody would know. Everybody would know it's a panda. If I asked 125 people, what organization is that panda linked with? You'll know it's a World Wildlife Fund. It's a beautiful, iconic logo. Now, the orangutan. The orangutan has become the symbol for looking at issues around deforestation for palm oil. And now more and more people starting to recognize the plight of the orangutan in rainforests associated with palm oil. Now, in my laboratory, we do lots of very strange things. And one of the challenges that I gave to my research group was, what is going to be our logo? How are we going to tell the world about our soya project? And we had lots and lots of different ideas and thoughts. But very recently, the winner was announced. And the winner of protecting the world's rainforest from deforestation from soya are these beautiful birds. They're called the hyacinth macaw. And I hope you will see these hyacinth macaws more and more to show the work that we are doing in trying to protect the rainforests from deforestation for soya production. I think it's also very fitting if we look at the colors of those beautiful birds, the blue and the gold. That's the colors of the Ukrainian flag. So not only are we going to support soya, but this is going to be the symbol of our solidarity with all of those people in Ukraine who are undergoing such difficult times at the moment. So I'm going to stop talking now, and I would be very, very happy to take any questions from any of the participants in the meeting. So thank you very much for your attention. And I think back to you, Doris.
Thank you so much, Chris. This was, as usual, a very inspiring talk. And I'm sure I talk here to everybody and out of the soul of everybody here. So many wonderful projects. And I don't see a hand raised yet. So, you may, there's the first question popping up. So, and the first question would be, from Maya, how sensitive are fingerprints to yearly season changes in growing conditions like drought, etc.?
That was from Maya.
Maya.
Maya, what a great question, because it's so important, the question that you ask. The types of databases that we generate, we call them living databases, because they always have to be updated. Every year we need fresh samples, because you know what? Climate changes. And as climate changes, the fingerprint of the particular crop in a region will change as well. So for our oregano project, believe it or not, we've collected 5,000 samples of oregano, and every year we'll update our database to make sure that it reflects the most recent crop. So Maya, well done, great question.
Yeah, there's one question I had also on my list, and a question of Daniel Reibel, and he asks about the GMO difficulty to analyze in the soy question. So do you address that, and how do you see changes, or do you include that at least in your studies?
Yeah, and again, it's another beautiful question. So what we are setting out to do, our project is very much about the origin of the soya. But GMO, non-GMO, is it organic, is it conventional, is also really important. So what we are doing is, for every sample that we are getting into our laboratory, we are trying to collect as much metadata as possible. Tell us about that soya, because once we collect that metadata, we can then start to interrogate all of the analytical results that we are producing. Say, yes, we can tell the country, but actually we can tell the difference between organic and conventional. We can tell the difference between GMO and organic. So that's exactly what we're setting out to do.
So do you get the information on GMO and non-GMO in the first place? Because you need to have the basic information to set up your database. So how do you make sure that you do have GMO or non-GMO?
Yeah, and again, that's a beautiful question, Doris, because what we do is we work with partners who can source the soya, and they know the supply chains. So they will know not only the country it comes from, not only the region, often they will know the farm that it comes from. Because if you just go out onto the marketplace and you buy stuff, it means absolutely nothing, really.
Exactly. Yeah. So the other question. So the next one is from Anthony Traszewski. Which of the analytical methods do you consider to be the most useful and versatile?
Well, Anthony, the answer is all of them. And that's why we've built our unbelievable technology center here. We have about 50 different analytical platforms, spectroscopy, mass spectrometry, sensor technologies, because sometimes you're never sure which will be the most appropriate technology. And we will always look for what's the simplest, what's the lowest cost technology, and how easy would it be to transfer that methodology? Because there's no point developing a beautiful test, which is an unbelievably complicated workflow that nobody will ever be able to reproduce. And so, for instance, in our soya project, That's why we went for volatiles, non-volatiles, and elements. We're not sure actually which of the techniques will give the right information. And it could be a combination of techniques. And then we will start to use data fusion, bringing data from different techniques together in terms of our model building.
Yeah, so the next question in the line is, did you apply your methodology to cheese?
Cheese.
Cheese.
Cheese is my favorite food, okay? I love cheese. And absolutely, yes, we've done quite a number of different projects in terms of cheese, because again, you go back into your supermarket and we pay a lot of money for different types of cheeses, particularly those that contain country of origin, a particular variety of cheese. So yes, cheese is a beautiful matrix, especially on the Reams technology, because it's particularly good at measuring the phospholipids, and cheese is full of phospholipids. So yes, absolutely.
Mm. So concerning the soy project, there's an interesting question from Annalena Meyer, and she would like to know whether if you can maybe determine whether soy is grown on deforested ground or not. Can you extend that to other crops like palm oil as well?
Yeah. And what we're doing is we're using soya as our model project around looking at innovations about where and how was it grown. Mm-hmm. And if I give an example, is we're already showing our data is very, very good at country of origin. We can pick that out already. And now we're saying is from the early indications, actually we're picking up markers of deforestation. And it looks like that's because there has been a lot of burning in the ground, and that burning material has entered into the soil. So there's absolutely no reason why the same could not be applied to other things like palm oil, like cocoa or cacao, for instance, exactly the same methodology.
Yeah, so the next question, I have to apologize, but the name is written, I think, in Greek. I can't read that, so sorry, but I can read the question. So from the data you have collected, can you discern changes of farming practices or environmental changes? So can you really follow the farming practices?
If in terms of farming practices, that is the separation of organic versus conventional, we hope the answer will be yes, but we're not sure yet. Mm-hmm. But actually using Reams technology, we've been able to do that in lots of different types of crop production. So we're quite confident about that. In terms of the environmental footprint, I think what we would have to do would be to collect samples over quite a long time period to see if you can use those measurements to measure environmental change. We really aren't sure about that.
Yeah. So, and a question from Guljar Armin is, by using this technique, can we know the fraud in other dairy products?
Yeah, so if you think about what is the potential fraud in dairy products, okay, and there's a lot of them. Just a short list. Is my milk organic? Is it goat's milk, or has it been substituted with another species? Is my oat milk or is my almond milk really made of oats or almonds, or has been substituted with something else? So there's lots of different questions there. And what we really believe is this idea of fingerprinting will give that unequivocal result in terms of, is it adulterated or is it genuine?
Mm. Then a question on the methodology from Anna Jill. Do you use NMR spectroscopy at all? It has higher reproducibility and easier and more rapid protocols than MS-based methods. And besides, it's more non-destructive, so you can reuse a sample for several other analyses.
Well, The short answer is no, we haven't used NMR spectroscopy. And I guess the reason for that is our background isn't really in NMR, to be honest with you. And what we have looked at is the speed of analysis and the ability to transfer methodologies between laboratories as well. And you know, we work with people who are very, very keen on using NMR, and often we will compare our LC or GC mass spec with NMR data as well.
Yeah. So I have to place one question, Chris, and so why oregano? Is oregano a model, or is it absolutely prone to fraud, or do we have to expect that type of fraud in every green spice, or let's say in every spice?
So why oregano was because that was the information that I was given, the intelligence. And it actually was because at that time there was a crop failure in oregano and the price was really rising.
Okay.
So what we did was, often for herbs and spices, we will model, we look at price information in terms of fluctuations. Does it go up or go down? And we can track fraud with those price fluctuations. Mm-hmm. And yes, it's not just oregano. Every spice we have looked at, every herb we look at, we find fraud, everything. Mm-hmm. So we're currently exploring fraud in ginger. Mm-hmm. And my God, we're finding lots of fraud in ginger.
Oh, really?
Yes, we're exploring fraud in turmeric, and we're finding a lot of fraud in turmeric. Mm-hmm. Now, for ginger, crop failures, price went up, we're finding fraud. For turmeric, the demand for turmeric globally has really massively increased because of all of these reports is that it helps you fight COVID. So the demand for turmeric has gone up. So we will look for a lot of economic indicators that will tell you that there's a likelihood of fraud happening, not only in a herb or spice, but in a particular type of fish or particular type of meat.
Mm-hmm. So one last question from my side. So what is the amount of additional stuff you need to have in the sample to detect something? You know, so I would expect, well, if it's an organically growing crop, you might have just a natural amount of additional stuff, which might be co-harvested. So how do you dissect between that and fraud? So when do you see it?
If we go back to the topic about organic, it's really interesting because we were not so sure that we would be able to tell the difference between organic and conventional produced, yet we're finding massive differences. And what we believe, the differences are around the microbiome of the soil. Because as you start to add chemical inputs, you're actually changing the microbiota in the soil. So you're changing the whole ecosystem. Therefore, the nutrients that the plants can absorb or can't absorb will be very, very different.
So can you say a percentage of byproduct you need to then have the possibility to sense additional stuff?
Yeah.
So is it 1%, is it 5%, is it 10%, or?
No, we've done a lot of work in terms of understanding how many samples do you need to put in to your database to produce robust data. And actually, we did a very large review of the scientific literature. Some people claim it's 5 samples, some people claim it was 50, 200. Now, what we really have reached the point is to say, if you don't have at least 100 genuine samples taken at different times from different areas, actually your statistics are not robust enough. So people can publish papers to say, using 10 samples, I found this and I found this. We actually go, that isn't robust science. You could not repeat that. You could not reproduce it. But again, we published work about spectroscopy and mass spectrometry in terms of the numbers of samples that you need to take, analyze, to build robust models.
Thank you. So more questions from the chat. So just a thought, it seems that portability of the analytical instrumentation is the future. What is your opinion there?
Oh, we love to think about portability of instrumentation. And again, Doris, a lot of what we've been doing over the last few years is looking at the miniaturization of spectroscopy. And what you can do is you can buy some really low cost NIR devices or mid-IR, and they can take the measurement. What you need is really, really good way to do data processing. And our solution to that is this. We will Bluetooth the signal from the miniaturized spectroscopy to our smartphone. The smartphone can then send that signature to the cloud where we have very complex databases. And as long as you've got a good Wi-Fi signal, within a few seconds, that database can be interrogated, come back to your smartphone, and it can be yes, okay, or adulterated. And we're doing a lot of work now in India, actually looking at adulteration of food products in India, just using smartphones attached to a very low cost sensor.
That's very interesting. So, and it's linked to the next question, saying from Judith, when shopping for groceries, is there anything oneself can do to not fall into traps of food fraud? So I think it's a little bit of future you have now considered. So what can we do at the moment?
I mean, again, it's a really good question, because if we think we have to walk into a shop or supermarket and do our own testing, that tells us we do not trust our food system. That's really very dangerous. Now, what I would always say to people is, if the cost of something, if the price looks too good to be true, it probably is, and don't buy it. And you should leave the rest of it to regulators, to the food business operators, to ensure that our food is safe and authentic.
So the next question I'm not so sure about. Someone wants to know about the link between soy and defrosting. So maybe someone wants, maybe Suela can comment a little bit on that. Maybe it's meant whether it's fresh or it was already frozen, if you can dissect here. This is how I understand it. I don't know.
There might be two questions there, and I will answer both of them. Actually, there is a lot of fraud about what is fresh and what has been frozen, thawed, and that's actually particularly around fish. Fresh fish, is it fresh, or has it been frozen 2, 3, 4 times? And again, you can tell that quite nicely through lipidomics, metabolomics, okay? If the question is not about the ability to tell whether it's fresh or not, but if it's come from deforested ground, again, it's about the profile of the molecules that you will find in that sample. Is it high in phosphates? Is it high on materials that would be linked to the burning of wood, for instance? Those are the sorts of markers that we would look for.
So, and there's another question on the oregano story, or not on the oregano. Did you have any experience in using bioactive compounds such as BACs from oregano or thyme? BAC, I don't know.
Yeah, it's interesting. I have a really good friend of mine, and he is a microbiologist. And often we argue to say, I say we use chemical fingerprinting, and he said, no, you should use microbiological fingerprinting. And we argue about it all the time. But I would say, actually, the chemical is a much, much better and faster way to do it. But of course, my colleague would say, no, microbiology is a better way to do it. It's just a different approach, biology versus chemistry.
And so the last question I see in my line is, what about sample treatment? Is it applicable for portable or in situ analytical instrumentation?
Again, really good question. There's no point of having nice, device to measure, send it to your smartphone. If you've got to spend 30 minutes doing sample preparation, it doesn't work. And also that sample preparation, does it include the need for solvents? Does it need and then you start to go, actually, it's not working at all. So a lot of our work has been around non-destructive measurement of samples, okay, where the only sample preparation is maybe grind it up in a little coffee mill, and then you take your analysis on that.
And the next question sticks to this analysis point. And could we apply this technique before we import any product from other countries? I mean, is it easy to apply, and does it cost a lot of money? We know that problem from the mycotoxin field, where many companies nowadays analyze before importing. So what would you say to that? So
again, we're really quite heavily involved in trying to answer that question, Doris. So we're working with a lot of companies, particularly in India, and they want to know, is it free from mycotoxins and is it authentic before they export it? Because quite often it would take 1 week or 2 weeks to get that analysis done within country. And that's a lot of time that you have to keep materials sitting there. So now we're working on all of this beautiful, portable, rapid technology, but we're also working on the digitization of supply chains, where you can get a QR code for a particular batch of material. You can do the scan in India, that then gets exported to Turkey. You can do the scan in Turkey, and you can track that material right across the supply chain. It makes it really difficult for anybody to cheat, to adulterate their product.
Yeah, that would be wonderful to achieve that. So now I have the impression that the questions, I have a lot of things in the chat which were wrongly typed, but I think that's okay here. So did I overlook any questions? I don't think so. So other questions from the auditorium? I don't see any hands raised. Wonderful discussion. So if I do not see other questions, so one last remark. I don't know whether you are aware, but I'm sure you are. You know that in Austria, GMOs are not welcome, and it's a big issue. The import of soy, not as GMO. So we really look forward to good methodology to really dissect between these products, and this is a huge market which has to be controlled. And at the moment, it's well, there are a lot of question marks on the material which are in the supply chains, and so that's a big topic actually. Yeah. So do you see a possibility when gene editing is coming into the market? How do you see your methodology in terms of dissecting between gene edited and non-edited products? Do you see a chance here?
Well, I think gene editing, I think, is going to be one of the really big techniques for this century, no doubt about it, to improve productivity, to make things more resistant to drought and salinity. And I think there's a huge number of positives about gene editing. But again, we need that transparency. We need to be able to make that decision. Do you want to buy gene edited foods or not? Now, again, I can only hypothesize in terms of our fingerprinting techniques. when you start just to take one single gene out of a plant or an animal, that is going to have a major effect on the metabolism, on the machinery, and we should be able to pick that up. In fact, in a much better way than, say, genetics would be, because if you're doing genetic testing, you need to know exactly which gene has been modified or removed.
Yeah. Yeah, maybe metabolomics is indeed then the technique, no? To trace that. Yeah, so and I have a lot of communication here saying, great webinar, great talk, many thanks for the great contribution. And this is really what I have to also say. A wonderful talk, very, very inspiring as usual. And I have the feeling I have to go to my shelf and look at the stuff, send you everything for analysis. And yeah, many, many thanks. Great talk.
If I can add some comment for the participants that are online, if they have some more questions, if they can send samples for your studies or so on, they have your contacts. So, well, I was hearing this and I was thinking I have to try to see if Portugal produces soya or whatever, because I want to contribute. So if any of the participants that are listening can contribute for this, of course, it's very important. And so one more time, I'll leave this request and they have your contact, and for sure they will reach you. So thank you very much. Thank you, Doris, for also for all the questions that you made that were very interesting. And for the participants, I have here also a lot of comments saying thank you, Chris. So thank you all. We hope in some months to organize the second webinar, and all of us, we will have to keep an eye in what is happening, and we have to have some hope that things will improve. And hopefully when we will meet in some months, the things will be better. So thank you all. Thank you for being with us. Thank you, Chris, one more time. And we will meet in a few months. So thank you. Bye.
Bye. Bye, Chris. All the best.